Hello, hi everyone. I'm Sandhya. I'm your botany lecturer. In the last video, we have studied about the biomolecules, introduction of biomolecules, and analysis of the biomolecules. Acid soluble portion, insoluble portion. Acid soluble portion is going to have biomicromolecules. Acid insoluble portion is going to have biomacromolecules. The introduction part we have studied. So in this video, we are going to study in detail about the biomicromolecules and biomacromolecules. Okay, so here biomolecules are divided into biomicromolecules and biomacromolecules. Okay, so micromolecules, we have come across. What are the examples? The sugars. Okay, sugars. First we'll study sugars. Sugars, amino acids, fatty acids, and nucleotide bases. Nucleotide bases. Okay. Let us study in detail about them. Sugars are nothing but they are the building blocks of the carbohydrates or the polysaccharides. Okay, or the polysaccharides. The polysaccharides are going to come under the biomacromolecules. Okay, these sugars are simple molecules. They are simple molecules of carbohydrates which are going to have carbon, hydrogen, and oxygen. Okay, they are monosaccharides. Okay, monosaccharides are going to bind to form into disaccharides, then to polysaccharides. Monosaccharides example we can have glucose, fructose are the examples. Disaccharide example lactose, sucrose are the examples. Okay, polysaccharides are nothing but group of monosaccharides. The monosaccharides are going to combine to form into polysaccharides, and these polysaccharides are going to have a bond which is called as glycosidic bond. With the help of the glycosidic bond, they are going to form into polysaccharides. And the examples of polysaccharides are nothing but cellulose, starch, inulin, glycogen are the examples of polysaccharides. If you take glycogen, it is an animal one. It is present in animals. Then cellulose. Cellulose is a homopolymer of glucose. Cellulose is a homopolymer. Means throughout the structure only glucose is present. One monosaccharide is present. Homopolymer of glucose, cellulose. And this cellulose is present in plant cell wall, paper, and in cotton cellulose is present. It's a homopolymer of glucose. Starch is nothing but it is present. It is a storehouse of energy in plant tissues, and it is going to attain blue color when iodine is added to it. Okay. Then inulin is a polymer of fructose. Glycogen is present in animals. Okay, the sugars are nothing but a simple one, micromolecules, where these sugars examples are monosaccharides. These monosaccharides, the one saccharide and the saccharide, they bind together, they form into disaccharide by a glycosidic bond. Then di will form into trisaccharide. Like that, many will be forming into the polysaccharide. Poly means many. Okay, they form into polymers. These are simple, whereas these polysaccharides and biomacromolecules are complex. Okay, this is about the sugars which are biomicromolecules, polysaccharides, which are biomacromolecules. Moving on to amino acids now. Moving on to amino acids. Amino acids are the simple substance, simple form of proteins. Amino acids are nothing but the simple form of proteins. They are the building blocks of proteins. They are the building blocks of proteins. These amino acids are going to have amine group. They are going to have amine group and carboxylic group. Okay, they are going to have amine and carboxylic group. This particular amine and carboxylic group is going to attach to a carbon, which is called as alpha carbon. Okay, this is called as alpha carbon. This carbon is going to have tetravalency, no? So if you take a simple structure of amino acid, one of the bond is hydrogen, then carboxylic bond, then amine bond, and another one is the variant substituted compounds are present. Variant compounds are present is the R1. R is going to change a lot. This R can be hydrogen in case of glycine. It can be methyl in case of alanine, and it can be hydroxymethylated CH2OH in case of serine. Okay, these are the simple amino acids. Amino acids are those chemical substances which are going to have carboxyl group and amine group, and they are attached to a carbon. Then this carbon is a tetravalent one. So two valencies are occupied by carboxylic acid and amine group. Another one is occupied by hydrogen. The fourth one is nothing but the R. R is nothing but it is going to change. It's a variable group. It is a variable group. Then these amino acids are of 21 type. That of proteins are only of 21 type. Proteins are 21 type, but amino acids are many. Okay. Proteins are of 21 type, and the chemical properties and physical properties of these amino acids are due to amine group and carboxyl group, and also because of the functional group of this particular R. These amino acids can be acidic. They can be of acidic, or they can be of neutral. They can be of basic also. If you say acidic is a glutamic acid. Glutamic acid is a acidic form. Basic amino acids is nothing but lysine. The neutral amino acid is nothing but valine. Okay, these neutral amino acids called as valine. Okay, so then aromatic amino acids are also present. Aromatic amino acids, the rings, the aromatic amino acids are nothing but tyrosine, tryptophan, tyrosine, phenylalanine are the aromatic amino acids. Okay, then this is the general structure of amino acid. Sometimes this particular amino acid may contain positive group and also may contain the negative group. So this type of ion is called as Zwitter ionic form. Where the ion which is bearing both negative and positive charges are called as Zwitter ionic form. Okay, Zwitter ionic form. This is about the amino acids. Okay, this is about the amino acids. So these amino acids are of based on the giving the H plus ions or giving the OH or OH minus ions when they're not giving any ions. Based on that, they are divided into acidic, basic, and neutral. Based on the rings, they are divided into aromatic amino acids. Based on the availability also they are divided, like essential amino acids. Essential amino acids are those amino acids which we have to take through our diet. Non-essential amino acids are those amino acids which are prepared by our body. So this non-essential amino acids no need to take, but essential amino acids we have to take through diet. Okay, we have to take through diet. So non-essential amino acids which our body is going to make, while essential amino acids which we get through our diet. Then coming on to the proteins part. Okay, so these amino acids are going to combine with each other and form into proteins. Okay, these amino acid, they form into peptides. Okay, peptide they're going to form into polypeptides. The polypeptides together is going to form into proteins. What type of bond is present here? Peptide bond. Peptide bond is present here. Okay, these are also polymers of amino acids. Proteins are polymers of amino acids, and they are not homopolymers. Same amino acid will not be present. Different amino acids will be present. Hence, it is a heteropolymer. So proteins are nothing but they are polypeptides. And they are having a linkage of peptide bond. And each protein is a polymer of amino acid. There are 21 types of amino acids are present, like alanine, cysteine, glycine, proline, tryptophan, lysine. And these proteins are heteropolymers, not homopolymers. Okay. Then these particular proteins, the most abundant protein in animal world. In animal world, the most abundant protein is collagen. Collagen is an animal protein which is present abundantly. Then when we compare both animals and plants, when we compare the whole biosphere, then the most abundant protein is ribulose bisphosphate carboxylase oxygenase enzyme. This enzyme is a protein which is abundant in the whole biosphere, plants and animals. In the whole biosphere, it is the most abundant it's the most abundant protein. Ribulose biphosphate carboxylase oxygenase. Ribulose biphosphate carboxylase oxygenase. It is the most abundant protein in the whole biosphere. Okay, these proteins are of four types. I mean, for structural point of view, if you say, these proteins can show primary structure. Based on structural point of view, they can be of primary, like serine, lysine, leucine, tryptophan, tyrosine. Like this is a primary structure. If you talk about secondary structure, they will be like this. Okay, so if we enlarge this particular portion, is nothing but the primary structure, but this is the secondary structure. They can exhibit the tertiary structure also. Tertiary structure like this. This is the tertiary structure. They can exhibit the quaternary structure also. The quaternary structure of protein is present in human hemoglobin. The human hemoglobin is a quaternary structure of protein. Okay, so this is the primary structure, this is the secondary, this is the tertiary, and quaternary structure. Quaternary structure is also present. Tertiary structure is nothing but the 3D dimensional value is the tertiary structure, and this tertiary structure of protein is absolutely necessary for the biological activities of the proteins. So tertiary structure is very important for the biological activities of the proteins. This is about proteins now. Now moving on to the fatty acids. Moving on to the fatty acids. Fatty acids are nothing but they are going to have, see, amino acid is having amine group and carboxylic group. Okay, fatty acid is also going to have carboxylic group. At the same time, the R group, R group is also going to be present. Okay, it's nothing but simple fatty acids. Fatty acid is a carboxyl group attached to R. So this is nothing but the general fatty acid. This is going to have a chain of it. 1 to 19 carbons are going to be present. 1 to 19 carbons are going to be present if you say about fatty acid. Okay, so if you take an example, palmitic acid. Palmitic acid is going to have 16 carbons. 16 carbons including the carboxyl carbon. If you take another example of arachidonic acid, it is also a fatty acid. Arachidonic acid, it is having 20 carbon atoms. 20 carbon atoms are present, including the carboxyl carbon, Then if you take another example, glycerol. Glycerol is nothing but trihydroxypropane. It is just like this. This is glycerol. So fatty acid and glycerol are the simple forms of fats. So fats are nothing but, the lipids are nothing but, they are the complex molecules of fatty acids. Fatty acids and these lipids are water insoluble. Mainly they are water insoluble. And these fatty acids can also be divided into saturated and unsaturated one. The saturated fatty acids are those which are without double bond. Without double bond are saturated fatty acids. The unsaturated fatty acids are those which are having the double bond. Other examples of fatty acids are based on the presence of this glyceride. These fatty acids are divided into monoglycerides, diglycerides, and triglycerides based on the number of these glycerols. Okay, then so fatty acids, monoglyceride, one type of classification. Other type of classification based on the presence and absence of double bond, saturated and unsaturated fatty acids. Then these fats are different with that of oils involved in melting point. The oils are going to have lower melting point. The oils have a low melting point. That's why in winter they are in oil only. There is in oil only. Another fatty acid can be the example of phospholipids. Phospholipids play an important role. They are the chemical composition of cell membrane. Then lecithin is also there. Cholesterol, all are the fatty acids. So these fatty acids and glycerols are the simple structures of lipids. Okay. So we can say less glycerol. These are the simple structures of fats and lipids. These are water insoluble. Okay. Coming on to nucleotide bases. Okay. These particular things are heterocyclic rings. They are going to have nitrogen bases. Okay. The nucleotide bases are going to have nitrogen bases. These nitrogen bases are divided into two types: purines and pyrimidines. Purines are nothing but, examples of purines are adenine and guanine. its examples cytosine and thymine and uracil also. Okay, these are the examples, nitrogen bases. Then these nitrogen bases are going to combine with sugars, which is nothing but ribose sugar, which is a monosaccharide, or deoxyribose sugar, and form into nucleoside. Ribose sugar is present in RNA and deoxyribose sugar is present in DNA. So what are these RNA and DNA? They are the nucleic acids. So these nucleic acids come under the biomacromolecules. Nucleic acids. These are nothing but DNA and RNA. Genetic materials. Mostly the DNA is a genetic material. Okay. So nucleoside. Then this nucleoside is going to combine with phosphate group or phosphoric acid and combine to form nucleotide. These nucleotides are the building blocks of the nucleic acids. So these nitrogen bases, sugar, nucleoside, nucleotide are the simple forms. These nucleic acids are the complex forms. These form the biomacromolecules. These form the nucleic acids form the biomacromolecules. Okay. Then if we take about the particular thing, adenine has a nitrogen bases. So adenine plus ribose is going to form into adenosine. Guanine plus ribose is going to form into guanosine. Cytosine plus ribose, it is going to form into cytidine. Thymine plus thymine. Uracil plus ribose sugar is going to form into uridine. Okay, so this is the nitrogen base. This is the sugar. This is nothing but the nucleoside. Okay, ribose is present in RNA, no? So that's why ribose is present in RNA, like this. And in RNA, instead of thymine, uracil is present. Whereas in DNA, if you take, it is going to have thymine. It's going to have thymine. And this nucleoside is going to bind with phosphoric acid. It is going to result into nucleotide, which are the building blocks of nucleic acids. Okay, so adenosine is going to form into adenylate acid. This is going to form into guanylic acid. It's a nucleotide. This is going to form into cytidyllic acid. This is going to form into uridylic acid. Okay, this is about the RNA. Okay, if we say about DNA, instead of ribose sugar, it is going to have deoxyribose. So deoxyribose. Then here it is going to be deoxyadenosine, deoxyguanosine, deoxycytidine, instead of uracil, it is going to have thymine. Thymidine plus phosphoric acid, thymidyllic acid. Like this. This are the building blocks of nucleic acid, DNA and RNA. So now how they are arranged. Let us see how they are arranged. This is the ribose sugar. This is the, here this is the ribose sugar, okay? This is the ribose sugar. This is the first, second, third, fourth, fifth. Ribose is a hexose sugar. Okay. Then here at the first carbon atom, the new nitrogen base is going to attach. Suppose if it is adenine. Okay. Suppose it is adenine. Then it is adenine. So nucleic acids. Suppose if you take a DNA. DNA is a double helical structure, isn't it? Like this, it is going to be present. It's a double helical structure. So here the nucleotides are arranged to form one particular chain. Then again, here the nucleotides are arranged to form another particular chain. So one chain of nucleotide to the other chain of the nucleotide are bonded together by the hydrogen bonds. So this is going to bind with the double bond with thymine, like this. Okay, this is thymine. This is 3 prime, this is 5 prime. Okay, so two chains are at one particular, they are intact because of these hydrogen bonds. Adenine with thymine, and here guanosine with cytosine. So these are hydrogen bonds. Adenine, thymine, double hydrogen bonds. Guanosine and cytosine are triple hydrogen bonds. Okay, triple hydrogen bonds. Like this, they are going to be arranged. Then at the third carbon atom, here the phosphodiester bond is going to be formed. Here the phosphodiester bond is going to be formed. OCH3 of another nucleotide. So this particular thing is nothing but the phosphodiester bond. Phosphodiester bond. Okay. Okay. So if we take sugar with nitrogen base nucleoside, okay, then this nucleoside with phosphate radical nucleotide. This nucleotide is going to attach to another nucleotide, this nucleotide. Then it forms a dinucleotide to trinucleotide, like that polynucleotides. So polynucleotides are going to form into nucleic acids and matter. So this whole structure, if you see, if we take this particular portion, it is a nucleoside. Then this nucleoside is going to bind with this phosphate radical. This is a nucleotide, like this one nucleotide to another nucleotide, dinucleotide to polynucleotide, like that. The whole chain is going to have polynucleotides. Like this, in DNA, one more chain is present which runs anti-parallel to it. Okay, it is a double helical structure. Now two strands are present and they are running anti-parallel to it. Okay, and double helical structure. So they are, the DNA is double helical structure. They are running anti-parallel to it. Two strands are present. Okay, and these two strands are intact with the help of these bonds, hydrogen bonds between the nitrogen bases, like this. Okay. Again here G triple bond C This is going to attach to another deoxyribose like this. Here again the bond is going to be present like this. These are two chains, two strands are not. Okay. Let me make it clear. So sugars, it can be deoxy or it can be ribose plus nitrogen base nucleoside plus phosphoric acid is nothing but nucleotide. One nucleotide plus another nucleotide is formed dinucleotide. Dinucleotide with another nucleotide, it is going to form into polynucleotide. Like this polynucleotide, it is forming into dinucleotide. Dinucleotide plus again a nucleotide, it is forming into polynucleotide. This polynucleotide is going to form into one chain. Polynucleotide chain, one chain. Okay, like that another chain is present, two chains of polynucleotide is forming into the DNA. Like this the DNA is formed. Deoxy sugar. Okay, like this. This DNA is a nucleic acid, is a macromolecule. If it is the RNA, here ribose sugar will be there, nitrogen base uracil is going to be present like this. Thank you.